Hi, I'm Karuna Shrestha, a PhD student at Teagasc Forestry Development Department in Karlsruhe, registered with the University of Göttingen in Germany. I'm working on ElmAsh Project funded under the Teagasc Walsh Scholarship Program. This project focuses on conserving and improving two important native broadleaf species, elm and ash. My research has three main areas of focus. First, I work on the micropropagation of disease-tolerant ash trees. Growing ash from the seed can take up to 2 years and introduces genetic variation. So, to produce the identical clones of the tolerant trees, I use micropropagation, a technique where small pieces of plant tissue called as explants are grown on a nutrient-rich media. These form rapidly developing cells known as callus, which then regenerates into a new plantlets, true clones of the original tree. Secondly, I study the microbiome of tolerant and susceptible ash trees to identify potential biocontrol agents. Research shows that tolerant ash trees often host beneficial microbes that can help control the ash dieback pathogen. Finally, I work on the elm conservation, focusing on the native wych elm and other elm species that have survived the Dutch elm disease epidemic. This disease, caused by the fungal pathogen and spread mainly by the elm bark beetles, has devastated elm populations across Europe. Through a citizen science initiative, we invite people to report healthy elm trees. So, we are looking for a tree which is above 5 meters tall and around 20 centimeter diameter at breast height. So far, we have recorded 67 such locations and established an elm gene bank here at Teagasc Oak Park, containing 41 putatively resistant elm trees from across Ireland. Our next step is to test their resistance to restore elms in Irish landscape.